So, over the next 2 days, I'm going to be giving very short presentations about the clinical trial landscape in genetics. Let's start with genetics first. So today we'll be focusing on two genetic variations, GBA1 and LRRK2. So let's just recap very, very quickly. Genetics of Parkinson's. So about 10 to 15% with people with Parkinson's will have a family history of the condition. And variations in GBA1 and LRRK2, as well as all the other genes on the screen, have been linked to an increased risk of developing. So this lovely wheel that my colleague Caitlin had produced. And this shows all of the clinical trials that are in the pipeline for Parkinson's disease. So on the top here, we can see the disease-modifying therapies and then the symptomatic therapies. So today we'll be focusing on the ones that are shaded in. And they focus on GBA1 and LRRK2. So let's start with GBA1. So GBA1 gene is the most common genetic risk factor linked to Parkinson's disease. So about 5 to 15%, the numbers vary, but about 5 to 15% of people will have some variation in this gene. And this gene is associated with the enzyme GCase. It's really hard to pronounce. So I'll just call it GCase. So this enzyme helps in the cellular breakdown. Oh my God. Okay. Helps the cell breakdown and disposing of waste. So like Thomas said in his presentation, there's this garbage disposal system happening in the cell, and the GCase enzyme is very important in this process. So in general, in people with Parkinson's, both with GBA1 variations and without, there are lower GCase levels. This means that the garbage disposal system is not working as well. So any type of toxic waste, for example, alpha-synuclein aggregation, like we spoke about before, is not being able to be cleared as easily. So this is thought to contribute to the development and progression of Parkinson's with people who have this genetic variation. So let's look at this. We have ongoing clinical trials targeting the GBA1 gene, one in Israel, one in the Netherlands, one in the UK, Italy, Canada, and 2 in the US. So let's focus on Ambroxol because 5 out of 7 of these trials focus on Ambroxol. Ambroxol is a cough medicine, which is being explored for potential use in Parkinson's. So the reason why people are very interested in Ambroxol is because it is thought to increase GCase activity, the enzyme that I mentioned previously, which is thought to be lowered in Parkinson's, in people with Parkinson's carrying the GBA1 variation. So researchers think that Ambroxol would increase this activity, increase the garbage disposal system efficacy, and therefore possibly slow the progression of Parkinson's or improve symptoms. So let's start with ASPRO-PD. This is Ambroxol to slow the progression of Parkinson's disease. It's a phase 3 trial. It will be happening in London at University College London, UK, and throughout the whole of the UK. This is a 2-year phase 3 clinical trial. It's a large one with 330 participants, and roughly half of them will have GBA1 variation. This is to check whether or not Ambroxol would be specifically efficient in this population as compared to people who don't have this variation. Okay, and then we also have 2 other phase 2. We have 2 phase 2 studies on top of that active right now. One of them is happening in the Netherlands, in Groningen. It's a 1-year study with 80 participants, and its primary objective is disease modification. And we have another one happening in Israel. The pharmaceutical company are testing Ambroxol, high dose Ambroxol specifically, in newly diagnosed GBA1. And then we have 2 trials. One of them finished earlier this year, targeting the effects of Ambroxol on cognition. So there's one trial in Italy and one in Canada that has just finished, just to see whether or not people who have GBA-associated Parkinson's whether or not this would help in the cognition symptoms and Parkinson's disease, dementia, and the Canada trial as well. There are other approaches to targeting GBA1 that don't involve ambroxol. There is a phase 2 trial of this chemical here by Bio. It's in the United States. It is a novel GCase activator, so it is thought to act very similar to ambroxol, but in a this is I think a small chemical entity. So they've produced something very similar to ambroxol, and they're going to be trialing it in 237 people with Parkinson's, and some of them will carry the GBA1 variant. And then there's also the early phase 1 to 2A trial of this chemical here, provided by Eli Lilly. And what they're doing is they're doing gene therapy. We're going to be talking about gene therapy later on tomorrow, but this is just to say that the gene therapy approach is also being studied in this field. And this will be a 5-year study. A small one, just to check whether or not it's safe and tolerable. But yes, it's active this year. And now let's quickly move to LRRK2. This is the other very common genetic risk factor for Parkinson's. It is present in about 1% of people with Parkinson's. As you may recall, GBA1 is 5 to 15%, so this is slightly less common, but still quite common. LRRK2 is a special type of enzyme, and again, it does help with the recycling of waste, kind of like GBA1. It has other it helps in other things too, but variations in this gene are actually thought to the overactivation of LRRK2. So as you may recall, in ambroxol, there was a decrease in the activity of the enzyme. This variation leads to an upregulation, meaning that the system becomes overactive and a bit more, too overzealous. So some evidence suggests that LRRK2 may also play a role in the formation of Lewy bodies. So for this, as of this year, there's only 2 trials active. One phase 2 trial by Biogen, trialing their newest LRRK2 inhibitor. So this inhibitor would block LRRK2 activity, hopefully lowering it to the desired level. And they are also trialing a different molecule, again, Biogen, with a phase 1 trial. I actually forgot to mention that all of this data is as of January 31st, 2024. So we have done this analysis as a group. It's people, Parkinson's research advocates, some of the staff at Cure Parkinson, some of the staff at MJFF. Yearly, this is a paper that we produce, and this data is accurate as to 31st of January 2024. So there might be a few months of delay. Perhaps there were some studies that have started up over the past few months that this analysis did not cover, but there's going to be another analysis coming next year that will then cover all of that. Okay, do you have any questions?
So how are you monitoring the results? Is it that GCase, can you monitor that? Or is it just by their motor symptoms improving?
I think the primary objective, the primary outcome of the clinical trial is usually the progression of motor symptoms. But there are some secondary and exploratory outcomes that some trials will be trialing, which is measuring the activity of GCase.
So you can measure that.
You can measure the GCase?
Yes, you can measure GCase activity. I'm not entirely sure how they do that in the
Well, this brings Anaya in and what we're going to be talking about this afternoon.
Okay.
Unfortunately, the only way we can measure it currently is using cerebrospinal, I can never say that word,
Yes.
fluid.
Yes.
So that involves a lumbar puncture,
Mhm.
and from that we are able to do chemical analyses with that, very vital fluid.
And then just one other thing.
On the cough medicine, so you had one study looking at cognitive, one looking at motor skills. Why wouldn't you have a study that looked at both? I mean,
well, a lot of studies will use a combination of different things, but regulators, and I'm going to bring Martin in on this because he's much the best placed to comment on this. Regulators tend to like trials to have one endpoint, one clear endpoint, not multiple endpoints. So it's a bit of a challenge. What we try and do in a clinical trial is collect as much information as we possibly can, and then have lots of different analyses that inform the picture, even though you have to choose one point that you have to measure at the end. Martin, was that right? Or...
You're absolutely right.
Hooray!
And so it also depends on what they call the mechanism of action, right? So you make a theory as to if I give this drug, then this might, be the result of it. My motor symptoms might improve, or my cognitive symptoms might, slow down, et cetera, et cetera. So you really got to focus on what is it that the drug particularly does? And that's where you build your endpoint around. Hopefully, you always hope that your primary endpoint works. And then your secondary endpoint, other things that you think might happen, such as cognition, work as well. But you have to focus on something.
Thank you.
Okay, so a layperson's question here, way backing up. So I'm listening to all of this and I'm thinking, okay, having traveled with a person with Parkinson's now for a couple of years, and in those clinical consults, the initial ones, usually the question that gets asked first is, do you have a family member with PD? And I think that's the reason why many of us with Parkinson's people in our life don't even know what our Parkinson's people genes are in relation to this, because we'll probably say, no, I don't think so. But then all of a sudden, years down the road, you say, hey, what about Uncle so-and-so? I think that could have been. But what's the base of our gene knowledge right now in terms of how many Parkinson's patients percentage-wise do we know are in the gene pool? And clinical trials that are, yet to come in the future certainly need candidates that have these genes. So what's the pathway to get your patient gene tested?
If you wanna-
So there are a couple of things that immediately draw your attention to. The first of which is Parkinson's Foundation's PD generation study, which is a very comprehensive genetic testing study that's happening particularly here in North America. So if you want to find out more information about that, then I'm sure that Parkinson's Foundation in Michigan will be able to guide you and lead you in that direction. You're about to hear a little bit later on today about the MIND study that's happening here in Grand Rapids, which is also collecting a lot of biosamples. And that is another way that information is being aggregated. There is also a fantastic effort that is being driven by the Aligning Science Across Parkinson's initiative, which is called GP2, which is about a massive genetic data, bringing it all together, putting it all under one roof. And we're all the time trying to make sure that any study has very broad consent so that that information can be shared and those learnings applied across the whole field. Jessie, is there anything else that I've forgotten on what you know? Because Jessie is a real advocate for genetic collection and very, very knowledgeable.
Yeah, I think you mentioned all the ones that I was going to mention. PD generation is very, very good. They have to have counseling, and we'll look at seven genes. And if they don't find it in the 7 genes, they'll do a whole genome sequencing. Because I think there's lots of papers now where they're finding more genetic than what was previously thought. And now, because some of these genes aren't highly penetrant, or people might pass away from other things before they had Parkinson's diagnosis. And what they're looking at is, like, I think there was a paper that just came out, like, maybe not first degree relatives, but second degree relatives. People don't really think about, well, my uncle or something might have had it, and maybe not my parents. So there's a lot of good research now on that. But yeah, I think PD generation, my dad did PD generation, is a really good study. And I've done ROPAD. And so there are some other things that you can get involved in. And I highly recommend everyone to get genetic testing, because the more data that we have, the more that we can figure out and have it in the.
And I think particularly if anyone gets Parkinson's, at a younger age, that is vital, absolutely vital, because that information has not been mapped very fully yet. I think the other thing that I wanted to say about GP2, which I think is incredibly important, is that they are building cohorts in Africa, in India, in South America, in Asia as well. And that means that we're starting to get a global picture of Parkinson's, because for too long it's been studied as a European and North American disease, and clearly it's not. So trying to understand if there are genetic clues that have happened through the endless diaspora as people have migrated, and really trying to understand the impact of that is going to be so important.
Curious a little bit about if you're genetically predisposed to Parkinson's, but you also are exposed to environmental factors. Like I know here in West Michigan, there are a lot of Superfund sites very close to where my parents have lived for the last 40, 50 years. And my dad was recently diagnosed with Parkinson's last year. And he hasn't had genetic testing done yet, but that's one thing they're going to talk about with his physician, Dr. Hedeman at Trinity next week. If you have a genetic predisposition, but I know you're saying not everyone then gets Parkinson's, even if they have those predispositions, but you also have that environmental exposure, does that increase your chance of expressing Parkinson's?
Yes, basically. So I don't know if I have a microphone. I'll shout loudly at you. But basically what we think Parkinson's disease risk is sort of a combination of multiple things. So it's not just, aside from the very few single mutations that drive disease, your risk of getting PD is probably from 3 or 4 or 5 or 6 or 7 different hits that have happened to you over your lifetime that have sort of slowly increased your risk until you pass some kind of threshold, and then the disease process starts. So environmental exposure combining with genetic factors, like perhaps if you had maybe not a gene, but a lot of those smaller, very common single nucleotide polymorphisms or these little mutations, you had a few of those, and then environmental exposure, that can kind of be a recipe for developing disease.
And adding on to that, then, it's my own personal theory, I guess. And I've seen a little bit of research being done on the correlation between COVID and Parkinson's and Parkinsonism. My dad really hadn't expressed any kind of signs or symptoms up until the last couple of years. He did have COVID at least once that we know of for sure. He was asymptomatic, had other issues that manifested weeks later. So I guess I'm curious too, are there more studies being done on that correlation between COVID and Parkinson's?
I mean, I think the initial sort of correlation we saw was after the 1918 influenza. There was potentially a surge of Parkinson's that they thought could be related. So there's been a long-standing hypothesis that infection may have a relationship to developing Parkinson's disease later in life. There's a few viruses they pointed out that people could have, just Epstein-Barr, things like that may increase your risk. There's a lot of studies being done on it. COVID specifically, it's very new. It's very hard to understand. There were like a very rare few handfuls of cases, like 2 or 3 or 4, where people had COVID and then developed Parkinsonism quickly after. But those seem to be very acute, very isolated events. I wouldn't be surprised. I mean, certainly the inflammatory properties of COVID didn't help anyone who's developing Parkinson's disease. It certainly didn't cause any benefit. If it's enough alone to trigger PD, I'm not sure, but we think it is probably, again, another one of those little hits that you can get across your lifetime that will kind of add on to your development.
Thank you.
Yeah.
Fantastic.